our DNA. Indeed, deoxyribonucleic acid has been one of man's most intriguing and interesting discoveries, and its chemical complexity and its immense potential in healthcare has always bewildered and fascinated scientists. I, Vaibhav, take immense pleasure in welcoming the respected dignitaries, heads of the administration, professors, and heads of various departments, chiefs, associate professors, assistant professors, postgraduates, and MBBS students who have all taken time of their busy schedule to be here today to the World DNA Day celebrations on behalf of the CCMGTR Club. Our club, which was established, which since its establishment has nurtured several young minds and made immense strides in the field of genetic research, is truly honored to host this wonderful occasion. Without further ado, let us now begin today's proceedings by paying homage to our motherland with a rendition of the Tamil Thyagarajan. I request everyone to kindly rise for the same.
The dignitaries and professors on the dais to kindly light the Kuthuvilakku to officially commence the event. Our Dean of Govt Kilpauk Medical College and Hospital, Professor Dr. K. Narayanasamy Sir, Vice Principal, Professor Dr. Ratnamanju Sri Jayaraman Ma'am, Medical Superintendent, Professor Dr. B. Aisha Shaheen Ma'am, Faculty in charge of CCMGTR Club and Professor and Head of the Department of Biochemistry, Professor Dr. V. Meera Ma'am, Joint Faculty in charge of the CCMGTR Club and Professor and Head of the Department of Microbiology, Professor Dr. Thyagarajan Ravinder Sir. Graciously invite Dr. V. Meera Ma'am to officially welcome the gathering.
A very pleasant morning to one and all. Genes are like the story and DNA is the language that the story is written. This is by Saint King, and the amazing you. The unique you is made up of this DNAs. So on this DNA celebration day, on behalf of the club, I just welcome each one of you. First of all, I must welcome our respected and beloved, dynamic, actually I'll be telling sir, really, literally he will be flying. We cannot catch him. So today morning 6 o'clock he started his rounds. And definitely it is a great inspiration to all of us, sir. In spite of your hectic schedule, you, sir, instantly accepted. I was just telling a word, he told I am very ready Anytime, any day, I am here only. So thank you, sir. A warm welcome to you, sir, for encouraging and blessing our children by your gracious presence. And our warm welcome to our energetic medical superintendent, Shaheen madam, who always very happily, because I'm very proud that she's a KMC here, and seeing the wonderful uplift of the KMC, it is a real very nice happiness it gives to any candidate of the என்ன சொல்றது இது வந்து பிறந்த வீடு. So we are enjoying it here. We welcome you, madam, and we welcome our vice principal, madam, very energetic, and especially this event, not only this event, any event, she is very meticulous, and step by step she will be guiding us and the students. Thank you so much, madam. We welcome you, madam. We welcome our RMO madam, Professor Vani madam, for gracing this occasion, and I welcome all the professors and our especially Ravinder sir, who is the joint faculty in charge, who always no word to say. It shows his dedication and kindness, always comes to help. We welcome you, sir. I welcome all the professors, associate professors, assistant professors, and all faculties from Kilpauk and GRH, and the wonderful team behind Sudharshanan Balaji, who is the key child of this genetic club, and his team. Each one, no words to say, sir. All the children were on toes, sir. And especially the first phase MBBS students, yesterday we just told them, they were all here with a wonderful rangoli and everything. So we welcome everyone, and I thank the Almighty, my parents and our dean and all the administrative team and the children for giving me this opportunity to deliver this welcome speech. I don't want to take much of the time. Sir, you are waiting to, the children are waiting to get the awards from you, sir. Thank you.
Thank you, ma'am, for that warm welcome. We would like to also invite Dr. G. Vani, ma'am, RMO, Govt Kilpauk Medical College and Hospital. So, we would now like to humbly request the Dean of our esteemed institution, Professor Dr. K. Narayanasamy, sir, who has always been motivating and supporting us in all research and academic initiatives, to grace us with his words. Please, sir.
Good morning to one and all. I am very happy to participate in World DNA 2023. This is the first time I am seeing that we are celebrating that this World DNA. Such a things will be this. There is happening anywhere in the globe or in the country, I don't know. But the madam has been taken such an insight into bringing to our young, energetic, and our undergraduates as well as the institution to make I think it will be a good initiation as well as it will be taking to practical, not only for just theoretical, they also be in initiation, implementation, followed by a follow-up also is there. This is a wonderful program. I cannot imagine such a thing will be the first time in my entire services to the academic and first time it is really is very much needed to that should be. Every disease having its own genetic potential, that is will be this epigenetic or genetic something is there. So in that aspect so far you have not been well ahead in that gene therapy. So for basis of a gene therapy, genetic is one of the most essential part to have this. So I think will be this from the undergrad. That is only another wonderful idea to make that will be from the. That is only is very important initiation. Why that is madam as well as will be joined to microbiology as a biochemistry, they are doing as a thing to role model for the entire medical college, for every other medical college hospital. The KMC is be as a role model for such a good initiative, so that everybody can follow that. So everybody, that is only everybody is having their own potential, but bringing such a potential to the forum, it is need for that is a small catalyst, small stimulus will be required, sensory stimulus. That's all be. So that will be, it is taken care by, that will be every faculty, especially for the head of the department as well as the entire faculty of the institution. Not only that will be the entire faculty of the institution. Bring out such things will be this rare opportunity. They can be read at the textbook, the undergraduate, they can read at the textbook, but on ground reality, see that will be, this is a thing will be DNA, this is the genetic, that will be this very, very important. Practical things. Always we are expected some practical things more viable, more retention than your theoretical things. When I get into observed and made have long-term retention, that will be for which we need to have some basic visual, that is virtual or in the practical. Two items are very needed to have a long-term retention. So that initiation should be taken care by, that will be this, it is a biochemistry department as well as the microbiology department. Another thing I wanted to see that in their department, and this the Sudharshanan Balaji or something, you only that is having some the collaboration DPT, that is with the KMC or something will be this. We are taking the project. The DPT is Director of Biotechnology. Microfluid analysis for the renal stones. That it can be implemented easily. He himself invented such a things, brought it idea, and he have been applied for this. Will be appreciated because will be this Director of Biotechnology. The Director of Biotechnology DPT is, I came to know such a terminology only after my finishing of super specialty during my establishment of multidisciplinary research into Madras. That's only I came to know that. Otherwise, I didn't know about such a terminology. That is a extramural funding source. There is an intramural and extramural. There are two types of the funding agency. One is intramural within our things, institution or within our DMA like that. The extramural means other sources for so many ICMR like that will be the DSR, Department of Health and Research, ICMR, and other things. So DBT, he has got to be a grant, and they have been invented his own idea that may be practically applicable to everywhere. Not only we keep this is a institution where will be highly sophisticated room where will be inviting the patient to. It is not like that. He has designed such a things will be they can be practicable in the territory and small things. That's only has been taken such a. That is will be I asked that we really I have been shocked to be seeing such a wonderful initiation by in his mind is coming to this. Is all whatever things we are born having something you are doing that all comes to the institution. So என்னால இந்த கல்வி மையம் நம்ம இந்த காலேஜ் நமக்கு என்ன செய்தன, நாம் இந்த காலேஜுக்கு என்ன செய்தோம், what we have done for this institution. So he has contributed. Everybody is ready, everybody is getting through, everybody is getting a pass, everybody is cleared, everybody is getting a distinction, everybody is having a good clinician, everybody is having everything. Everybody is having everything. But the only thing should be this unique. Madam already emphasized many times. The unique, the nature. That's only is needed for everything. Whatever you are doing that is a conventional. We come to this will be modernized with having without what we are so far being last week we are discussing about will be the drug dosages and that will be this producing the drug be the lesser side effect. Similarly to that, which is the gadgets, we are creating, we are producing that will be this, which having a less, that is easily can do that with the simple methods, very simple methods, the small investment, so bring us create outcomes. That is only it is having I said when I am originating such a program. I am very happy. It's my lifetime. I must be honored to have come over here to have take part in such a wonderful program. That will be not as a dean, as a medical fraternity. I have come here to have a participation. But the need for there will be this further. I think it will be after the biochemistry after Ravinder, after Meera Krishnan. Everybody is having a, but the other faculty should follow to stimulate our undergraduate to put in the consistency of the program. I think it will be this Sudharshanan Balaji is a role model to have a create our among this similarly such all the scientists are there, all are here, all scientists are here, but they must have some idea to bring out such things. Everybody can, nothing is impossible. We can, everything is possible. Only need for your thinking. The thinking itself, 50% is over. Your innovation is over. The constructive thinking of your mind, 50% of your invention is over. So second part will be this, how to make that result. So I take this opportunity to congratulate to all that is the entire team working for this as a wonderful DNA, World DNA celebrations will be this and role model for the every medical profession. Should be as a continue. I think if they have might be collaboration with the various the other institutions, not only have a medical institution, all the other that is IIT and all other good reputed institutions, they have a collaboration so that we can have this common solution for the difficult critical solutions. I once again to the thank that is the undergraduate of the entire team of this today's program and madam and the biochemistry, microbiology and the entire the medical college of all the departments who have been every department, even the having a the anatomy they have their own biochemistry, pathology, all they have. This is a common subject. It is not particular pertain to only biochemistry. It is a common subject. It does not require to be a particular department. Even the non-medical, they have their diseases. So the DNA is not been. I have not been expertise to talk further in detail. You know, I had an opportunity to undertake a human HRD fellowship. I got the HRD fellowship, Human Resource Development Fellowship. At that time, I have undergone to have a 2 months training to have the same molecular genetics. I undergone the genetic training for 2 months. I practically did such all these things. But as it was 10 years before now, I have been exhausted to complete. This is only I didn't follow the further. But that should not be in the level curriculum. Should be that is the followers, both sides should be the teacher as well as the students must have continuously, persistently to take part in such things beyond your routine curriculum. Okay, so once again I'll be again because will be this is the first in my medical curriculum. I am attending such things. Nobody will get the opportunity as a dean, as a faculty, as a hepatologist. I got the good opportunity to join with the main stream of your World DNA, and it has been further proceeded. Thank you very much for that.
Thank you, sir. I humbly request our beloved Dean of Govt Kilpauk Medical College and Hospital, Professor Dr. K. Narayana Swamy sir, to kindly accept a token of our gratitude from Dr. V. Meera. I kindly request our Vice Principal, Dr. Ratna Manjushri Jayaraman ma'am, who has been with us through every step of the way, to now kindly accept a token of gratitude from Dr. K. Geeta ma'am.
Sir, to give the memento to our Medical Superintendent Madam, Maisha Sahil Madam.
We would like to request Dr. G. Vani ma'am, the RMO of the Govt Kilpauk Medical College and Hospital, to accept a token of our love from Dr. Thyagarajan. We request Dr. V. Meera ma'am, the faculty in charge of the CCMGTR club, professor and head of the Department of Biochemistry, Govt Kilpauk Medical College and Hospital, to kindly accept the token of our gratitude from the respected dean sir. We take great pleasure in inviting our beloved Vice Principal ma'am to kindly honor Dr. Thyagarajan Ravinder sir, joint faculty in charge of the CCMGTR club, professor and head of the Department of Microbiology, Govt Kilpauk Medical College and Hospital, with a token of our gratitude.
Before we move on to the most exciting part of today, the prize distribution ceremony for the various competitions held to commemorate the World DNA Day from May 2nd to May 4th, we take great pleasure in facilitating the judges of the various events, without whom none of this would have been possible. Professor Dr. V. Meera ma'am, Head of the Department of Biochemistry, Govt Kilpauk Medical College and Hospital. Professor Dr. Thyagarajan Ravinder sir, Head of the Department of Microbiology, Govt Kilpauk Medical College and Hospital. Professor Dr. T. Aruna ma'am, Head of the Department of Pharmacology, Govt Kilpauk Medical College and Hospital, the judges of the debate competitions. Dr. K. Geeta ma'am, Associate Professor, Department of Biochemistry, Govt Kilpauk Medical College and Hospital, the judge of the Just a Minute speech competition. Dr. M. Sugati ma'am, Associate Professor, Department of Microbiology, Govt Kilpauk Medical College and Hospital, the judge of the Rare Genetic Case Presentation Competition. Dr. A. Balasubramanian sir, Senior Assistant Professor, Department of Biochemistry, Govt Kilpauk Medical College and Hospital, the judge of the meme competition. Dr. E. Srividya ma'am, Assistant Professor, Department of Anatomy, Govt Kilpauk Medical College and Hospital, the judge of the artistry contest. Thank you, ma'am and sir.
We kindly request our Honorable Dean sir to felicitate the judges of the various competitions. Thank you.
Madam,
We request the dignitaries, our respected Dean Sir, Vice Principal Ma'am, Medical
we request our beloved Dean Sir to felicitate Dr. Sudharshanan Balaji Sir from Untitled.
We request the dignitaries, our respected Dean Sir, Vice Principal Ma'am, Medical Superintendent, Faculty in Charge and Joint Faculty in Charge to kindly award the winners of the various events. Debate on the topic, research. Is it researching or just redoing? First place goes to Deepika B. Second place goes to Aditya R. Iyer. Debate on the topic, animal models for research are needed even in this advanced era of biotechnology. First place goes to Ananditi Singh. Second place goes to Rishabh Jain. Debate on the topic, collaborative research, a platform to exchange ideas versus to steal ideas. First place goes to Saravana Kumar SK. Second place goes to Venkat Dadich.
kindly request Vice Principal Ma'am to kindly give away the following prizes. Just a minute speech competition. First place goes to Aditya R. Iyer. Second place goes to Saravana Kumar S.K. Third place goes to Rishabh Ji. We now request our beloved Superintendent Ma'am to kindly give away the prizes for the meat contest. First place goes to Sobana Ji. Second place goes to Shesharaj B. Third place goes to Sanjay R.
We now request our beloved RMO, Dr. G. Vani Ma'am, to give away the prizes of the artistic contest. The first place goes to Muthuvalli Ammai Saravanan. The second place goes to Gayatri Ji. Third place goes to Deepika P. Thank you, ma'am. We now request Professor Dr. V. Meera ma'am to give away the prizes of the rare genetic case presentation competition. The first place goes to K.K. Bhaskar Subramanian. Second place goes to Rohit S. Third place goes to Rishabh Ji. As per the judges' wishes, we would like to felicitate every participant of the rare genetic case report presentation for their effort and excellent presentation. We request Dr. Thyagarajan Ravinder sir to give away the following prizes. Roshan Fatima. Not here. Sarada Devi. Sridharan. Venkat Dadi. Narmada. Karthikeyan. And Vidyadas. We request the dignitaries to kindly take their seats on the dais. Thank you. The first year MBBS students of our institution: Vidyadharan M, P. Ramesh, P. Rajesh, Kugan T, Sesharaj, Sanjeevan, N. Yugneshwaran, Sridharan P. K.S., Rajan, Vignesh P., Marudavelan M., Sanjay R. and T. Karthikeyan.
Thank you all. It indeed was a pleasure watching them perform. We now request the winners of the debate session to present their arguments and counter-arguments on stage. We now invite Deepika, second year MBBS student, who won the debate on research, is it researching or redoing, to share her points.
Aditya. I'm Deepika from batch 21. My topic is to defend that redoing is more important in research. Research is wherein we explore data to create new novel breakthroughs, and these new discoveries always need to be upgraded and up to date. We cannot just settle for results of the past researches forever. We must use information from past researches as founding stones and redo it again at present times to see if past results still hold true. Redoing especially becomes very essential in the field of antibiotic resistance patterns, epigenetic researches, and when data is compared between different nations. In an ever-changing world with evolution in technological advancements, redoing yields precise and reliable data. I conclude by saying, let's not narrow our minds to the speck of past findings, putting our present and future at doom. Rather, let's understand how much redoing and research stands undisputably important. Thank you.
We invite Ananditi, second year MBBS student, who won the debate on in this era of advanced biotechnology, are animal models necessary, to share a few points with our best audience.
Hello everyone, I'm Ananditi Singh of batch 21 and I'm here to talk about the topic, in the era of biotechnology, do we need to use animal models or not? I strongly believe that we need to use animal models and I'll tell you why, but first of all, for what? We use animal models for two things. First of all, we use it for testing the safety and efficacy of a drug, and next, we use it to develop new vaccines or new antibiotics for a particular disease. But why animal models? Why not something else? This is because animal models have genomes that are very similar to our genomes, and that's why they're going to react similarly to a particular antigen or a drug when it's introduced into them, just like how we do. And when we do animal research, we need to take care of 3 points, the 3 R's: reduction, refinement and replacement. Reduction as in we need to use the minimum number of study animals that is required, not more, not less. Refinement, that is to use aseptic conditions for the research, and also to give anesthesia to the animal groups if the pain is, if there's going to be pain due to invasive procedures. Finally, there's replacement. That is, you do the animal model research only when it's absolutely necessary. That is, when you don't have any non-animal based study approach available for this. So I would like to conclude my speech by telling that animal models are absolutely necessary, and I'll ask you this question, that will you volunteer for a human drug research trial if it wasn't done on a living model previously? I won't, and I hope you agree with that. Thank you.
Thank you, Anand. We now invite Saravana Kumar SK, second year MBBS student, who won the debate on collaborative research, a platform to exchange ideas or to steal ideas, to share his argument in this forum.
A pleasant morning to the respected professors, respected seniors and dear friends. Collaborative research, the very concept of collaborative research states that several scientific minds from various disciplines, various hierarchical positions come and volunteer to bring to forward the progress of scientific research. But this ideal is a far cry from what has been happening, as collaborative research that I strongly believe is becoming a forum to steal ideas. I have heard personal anecdotes from my friends from various other disciplines that their ideas were hoarded, their ideas were stolen, and they were not respected and not given due credit just because they were juniors or lower down the food chain. I heard one anecdote from my friend who has had to go to an internship to which she was only one among the 12 selected, but she was not able to go because the principal would not allow her to go, and he requested, he wanted undue credit for things which he didn't do. Thank you so much.
Thank you, Saravana. We request the winner of the jam session, Aditya R. Iyer, third year MBBS student, to present his winning speech on stage. Thank you.
Hello everyone, I'm Aditya from the 2020 batch, and the topic that was given to me was gene sequencing, and I'd quickly like to summarize my points. Gene sequencing is the process of determining the nucleic acid sequence in DNA. Therefore, it includes any method or technology that is used to determine the sequence of the 4 nitrogenous bases that we know. That is adenine, guanine, thymine and cytosine. Now, the most commonly used method is a high throughput method called next generation sequencing, which consists of 4 steps: nucleic acid isolation, library preparation, clonal amplification and sequencing, and data analysis. Now, the knowledge of these DNA sequences has become indispensable for basic biological research, DNA genographic projects such as the Human Genome Project that we all know, and applied fields such as medical diagnosis in cancer, for instance, biotechnology, forensic biology, and even agriculture. Earlier gene sequences were determined using 2D chromatography based methods, but the advent of rapid DNA sequencing methods such as fluorescence-based sequencing and automated chain termination DNA sequencing has greatly accelerated the biological and medical research and discovery. The major roadblock to gene sequencing revolution has been the issue of bioethics, but several major countries have formulated acts and commissions to ensure proper regulation, paving the way forward for genetics as a whole. Thank you.
Thank you, Aditya. We request the winner of the Rare Genetic Case Report Presentation Competition, KK Bhaskar Subramanian, first year MBBS student, to briefly present his case on stage.
Good morning, respected seniors, teachers and all my dear friends. And I'm going to present my topic which based on the relations between Fragile X syndrome and precocious puberty. So basically the summary of the case was a 7-year-old female came to the obstetric OPD presenting with chief complaints of pubic and axillary hair growth and height which is above the normal, above her age is of 150 centimeter. And she had a family history where her maternal first cousin had a Fragile X syndrome. And other complaints were she had macrocephaly and the breast development was Tanner stage 4, whereas the pubic hair development was Tanner stage 3. So the examination was, we examined it and there was no abdominal pain or any on palpation. And when we took the abdominal CT scan, there were no abdominal masses. And when we took the MRI scan of the brain, there was no lesions or any tumors or any such sorts. And when we took the wrist X-ray of the bone, we found that it was 13 years of age, which is greater than what of her age was, which indicated that the bone had developed beyond her age. So using all this, we came to the diagnosis that it was isosexual precocious puberty, which was true precocious puberty, and it was important that we had to differentiate it from false precocious puberty. We could do this because we measured the hormone level of gonadotropin releasing hormone as well as the FSH level, which is elevated and which is not elevated in the false precocious puberty. And since her mother had complained that she also had symptoms of slight mental retardation and she was not able to focus in class as well, therefore we did a Southern blotting test as well as the PCR for her mother's gene as well as her gene, which we found that in the FMR1 gene present on the X chromosome, there was the CGG repeats was greater than 200, which is the limit. So there were 750 repeats in her, while in her mother it was only 100 repeats, which indicated that the mother had a pre-mutation, whereas the daughter had the full mutation. Therefore, she manifested disease. This also indicates that the Fragile X syndrome is passed via oogenesis, that is via the maternal transmission and not the paternal transmission. So the basic, I'll conclude with the pathophysiology of the Fragile X syndrome. So basically the FMR1 gene codes for the FMRP protein, it is a familial mental retardation protein. This familial mental retardation protein is present in the cytosol of the neurons in the brain. So this one goes to the nucleus of the neuron and it helps in carrying the mRNA from the nucleus to the axon and the dendrites. There, this mRNA helps in translation of other mRNAs, which helps in releasing various helps in the synapse of the neural signals, and therefore the mental retardation occurs. Therefore, we conclude that there was a association between mental retardation and precocious. Thank you.
Thank you, Bhaskar. We also have the runner-up, Rohit Hess, who will be briefly presenting his case on stage as well.
Good morning, everyone. I myself Ravinder of MBBS first year. My topic was a case presentation of an AIS, that is antigen insensitivity syndrome. The case was very rare. Like a 24-year-old male came with an OPD with her build was normal with a female phenotype, and her pubic and actually her asparagus and labia were poorly developed, and her vagina was not normal. So we diagnosed her. We found the karyotype was XY. So it was male karyotype with female phenotype. So she also had a complaints of primary amenorrhea and inability to conceive for last 4 years. So we categorized into 3 different diagnoses. So we found that it either can be a Mullerian agenesis, or it can be a gonadal dysgenesis, or it can be a AIS. So we found it to be AIS only after we found that the hormonal level of the testosterone was normal, and the FSS and LH was above the normal around 90 milli international unit per ml. Then we came to the diagnosis of it that it is surely an AIS. Then we went for the treatment. Actually the AIS is due to the defect in the AR gene. So we can't, the gene therapy for it is still under research. So we treated it with CAIS, categorized it with CAIS. There is a complete and a partial and a mild AIS, and the complete AIS was treated with the removal of testes as gonadectomy and vaginal dilation, and the PAS was treated on the basis of whether it was a male or female, and the male was treated with orthopexy and hypospadias repair, and the female was treated with gonadectomy and estrogen and androgen therapy. So at last, the psychological assessment should be there, because a male revealing of this case is actually a psychological case. So we gave them with educational resources for assessing the information on what the defect is about and all this. Thank you.
Thank you, Rohit. I now call upon the founder and the student president of the CCMGTR club, Sudharshanan Balaji, 2018 batch to MBBS student, to kindly address the gathering and throw light on the achievements of the club, its activities and its goals.
Respected professors, I'm very honored to have this opportunity, and at the outset I would like to thank the institution and the departments of microbiology and biochemistry for giving me this opportunity. So talking about this club is, I must say that only Meera ma'am pointed this out. It's the first of its kind across the world, apparently, and I came to know very recently. And we have 140 plus members. We started with 140 plus, and then we had a few more exits and so much more entry from this current batch. Then again we are back to 140, and we are conducting weekly meetings on various genetic topics, techniques, in a way that is comprehensible. It's more of a discussion rather than a talk, because we find that on advice of the faculty advisory board, we felt that to be much more efficient. Some of the achievements of the club. So I will first start with the awards. So the list is very long, so I will read the awards first and then the names later, if I may be allowed to. So Sai Ganesh of final year MBBS has won the first place in AIIMS Raipur and LHMC Delhi in paper presentation. Both were on forensic medicine and one was a genetic topic. We've had quiz competitions won by Sri Hari and Sarnath final year MBBS students in national level quizzes held at Chengalpet Medical College, Madras Medical College, Stanley Medical College, Christian Medical College, SRM Medical College and Sri Balaji Medical College, to name a few in this current year. And then we had first place in the debate competition in both Madras Medical College and Stanley Medical College. Stanley Medical College, we also won the second prize in debate. Then first place in poster presentation at Madras Medical College and special prize at Stanley Medical College.
Credits for all these debates and the presentations goes to Aditya R. Iyer, Navina Mailarasu, Sachin Sunil Nair, Roshan Fatima, Varun Mughil, Vipin and Sivanesa Pandian. In addition to this, as a part of Medinal in 2022, we were able to conduct an ARMS PCR workshop on identifying human mutations. And the workshop was well attended by participants, MBBS students from Madras Medical College, Omandurar Medical College, and we even had a student come from Coimbatore Medical College. In addition to all this, amongst the club members, more than 25 members were fully trained in ARMS PCR and now are principal investigators on fully funded projects from the central government on topics of bladder cancer, prostate cancer, recurrent renal stones, in addition to osteoarthritis risk prediction. And now we have recently started as both research and diagnostic tacrolimus expressor status CYP3A5 mutations for renal allograft recipients, based on which the drug dose is tighter to prevent graft toxicity. In addition to this, we are very happy to share that 31 ICMR STS proposals have been sent from KMC this year, which on retrospective analysis is the highest since 2012. Now, the World DNA Day competitions put me in a very tough spot because all the judges were continuously frowning at me, saying I put them in a very tough position to choose the prizes because they had such a tough job. Of course, Sugandhi ma'am. So thank you all the participants for such intellectually stimulating literary events. We planned it as a small thing and it grew into a big tree. We are also happy to share that we have received inquiries from JIPMER and other Tamil Nadu government medical colleges asking to open, and just yesterday I got a call from Hyderabad from the director of CDFD to ask if they can push their students into the club. And we have received critical appraisal from the director of AIMS Delhi, CSIR CCMD Hyderabad, Dean of Ashoka University, former director of CSIR IGIB, senior faculty and deputy directors of multiple ICMR institutes, AIMS Patna, National Institute of Biomedical Genomics, Indian Institute of Science Bangalore and IIT Madras. To educate and advance our scientific knowledge in India, we first need to be aware of what happened in the past. Unfortunately, as it has been the situation, we are very much unaware of the kind of scientific advancement that's happening in our own country. I'm just talking about the government sector here, and within the government sector, even within state or within central institutions, there's hardly much knowledge about what the other person is doing. So this was continuously probing me because a lot of the club members were asking for ideas. So I'm very humbled to share that I was able to connect with senior faculty, deans and directors, and we have prepared a short video called as Snippets from the Experts that we will be releasing today, which contains compilations of 1 or 2 minute videos of all the scientists talking about their work. And I'm very happy to also share that most of the institutes that I've listed earlier are willing to sign MOUs with Kilpauk Medical College and take their research forward. So, completing what Ravinder sir said during the inauguration, trying to bring from bench to bed. So the idea of this club dawned upon the fact that we had a batchmate who we always used to joke around. Extremely academically talented would be the right word. I think all professors will agree. We used to joke saying, "Ikkove IAS pair add pannidala," because she had planned till IAS. Unfortunately, we lost her because she chose to end her life, which really probed me to look into what happened. And recently, NMC also released some data which said 65 suicides from MBBS students in the past 5 years. Now, this made me understand a fact that the academically inclined audience in medical college is a small group. There is no platform to hone their skills, to interact with like-minded individuals. There is no academic sharing platform of knowledge that they have. And this group is not usually involved in sports and other activities also. As I think all the professors and students will agree, MBBS is a tumultuous experience. To fit in somewhere, to find something constant in a turmoil of changes in personal and emotional and academic domain is a challenge. That was the reason why we started this club, and we named it Translational Research, because translational research includes everything. Any kind of research that can be done comes under its domain. The 80 plus faculty, each of whom I had the honor of interacting with, each suggested few ideas. So this started as an ant-sized project and went on to become a mountain with all their ideas, with their multiple years of experiences. We did have setbacks. I would be lying if I did not deny that. Quite a lot of setbacks. But one particular faculty was pretty hesitant on the idea. But what that made us understand is that when 80 senior faculty have given their ideas and put the idea forward, it showed us that as long as there's constructive criticism from those who are not in favor of the idea, we must take that and move forward. And thanks to support with VP ma'am, Dean ma'am, and all the faculty, we were able to pull this across. Our faculty advisory board consists of all the chiefs from both Kilpauk Medical College, Roypeta Hospital and Otteri and another peripheral hospital. We have 80 plus faculty who have been guiding us out of their working hours in the middle of the night, and we are very, very grateful for that. So generally, when we talk about courts, for example, somebody files a case on somebody else. Now in civil courts, it's the onus of the respondent to prove that they are not guilty. So our court system unfortunately believes that you are guilty until proven innocent. So like that story, a lot of these setbacks helped us modify the club and bring it to where it is today. So I would be very grateful for the fact that we had hurdles, because if we didn't have hurdles, the club couldn't have succeeded so far. It helped us modify every small bit. We could pay attention to all the details and ensure that the club came out well. Generally in marriages, they say in sickness and in health, in good times and in bad times. Now I am unmarried, so I don't have any idea of how true it is. But I do know one thing, only mentors, professors, especially all those sitting here and who are not here, they don't take an oath in an altar. They don't walk around the fire, but they are always there for us. And that I think is the greatest thing that this club has given all of us. We went ahead with the idea to show that when there is experienced faculty, we can do anything. So that is what prompted us to go ahead. Even though we are a student-initiated club, I would be wrong in saying student-initiated if I don't add the word faculty-enriched club. So generally when we talk about true love, I have seen it only in two places, from parents, and the never-ending love and affection from all my gurus, professors and faculty. When we talk about love, I will not be doing my duty if I don't fail to mention the Natya Shastra that says there are 9 emotions. That's actually a misnomer, a wrong fact. There are only 8 emotions. Shanta or peace, which we think to be an emotion. Yes, it is, yes ma'am. So it is actually the most complex of emotions and an amalgamation of all of this. So the point here is that we can never truly know or understand what peace is. So that being said, what we do know is that a kind gesture amidst busy schedules, can change your entire life, your entire day. We are all here because of so many professors who have had a kind word to share with us at the end of a very troubled or a very difficult day. And that is the only reason I am standing here, and I'm sure most of my seniors, juniors and my batchmates will agree. Now, kind words work both ways. What do we lose to the students here? What do we lose by greeting a professor? What do we lose by, asking them how they are? We have no idea of the kind of stress the kind of battles they face, the amount of sacrifices they do, the amount of tough decisions they have to take, dropping their family, running for duty. Why do they do it? Have any of us bothered asking? There's a famous saying in corporates, it's lonely at the top. So let us give them back one drop of the ocean they give us, and let us just first respect them as elders and be kind and share a kind word. Now there are lots of lessons I've learned through this entire process, some which I wish to share here. Dr. P. Parandaman sir was always, the professor and head of medicine, was always talking about tracking of variants. So does anybody here know what the latest COVID variant is? No. Obviously there have been hundreds of variants, how can we remember? But the point of knowing variants, Parandaman sir will say, is very important. And this was proven when I had the privilege of interacting with the director of CSIR IGIP, who's in the WHO task force for emergency pandemic preparedness. And he told that only if you know what you are fighting, you can fight it. It's a very important part of public health preparedness to know what variant we are facing. Dr. T. Balasubramanian sir, professor and head of nephrology, had World Kidney Day celebrations where we all got the opportunity to meet Professor Dr. N.K. Mani sir, very inspiring person, who told one statement which made me rethink everything. He told, we are talking about dialysis care, enriching dialysis care. But dialysis itself shows that you have failed to prevent the onset of chronic kidney disease. So what are we doing here as nephrologists, he asked. Now as an MBBS student, I was very shocked because I've never heard senior faculty throw such alarming statements, and this made us understand that you need not work from diagnosis to treatment. Sometimes after treatment also we can come back to diagnosis. So working backwards, the reason why genomics is genomics today or genetics is genetics today is because we have failed in phenomics or identifying the phenotype. So we are trying to find the cause. Then why genetics? That is a question because we are still in the research phase. We have not gone into diagnostic mode of genetics entirely. So the point here is that biomarkers will always remain the gold standard. Genetics will just aid the biomarkers or phenotype or protein expression. Does anybody know when the whole human genome was sequenced? Students? What? 2006, 2003? All of you are wrong. The human genome was completely sequenced only last year, exactly 1 year and 1 month back. Till now, we hadn't sequenced the telomere regions. So telomere to telomere sequencing or gapless gene sequencing was done only last year. And that shows how much more we have to reach in genetics. When we talk about rare diseases, WHO defines it as 1 in 1,000. In a population as large as India, that is 1.4 million cases. 1.4 million cases, I repeat. So this is not rare. Next time some professor tells us some rare point or a rare disease, we might want to pen it down because we don't know with such high rates, we don't know when we'll face what case. There is a famous cookbook called Meenakshi Amman's Samaithu Park. Now cooking is a beautiful analogy to genetics, as all the professors who trained me keep telling. And the ingredient and the edathla pota, everything will come out. In fact, ingyu mixie da irukku. Centrifuge vortexes are adapted mixies only. So everything has an analogy in life, and when we analyze that analogy, we can get more things. So I was very fortunate to find a method of reducing the cost because I saw my mother putting less uppu and putting more sambar podi in the samayal that morning. I came and tried it with the primers and I was able to reduce the cost. I'm very grateful to cooking for teaching me medicine. Dr. Teres Mary ma'am, Dr. Suganti ma'am and Professor Ravinder sir always keep talking about phenotype versus genotype. So they kept telling me that even in HLA typing, like just day before yesterday madam was telling, the CDC or serological method is always advanced when compared to genotypic methods. Even in AMR, antimicrobial resistant policies nowadays, we are slowly incorporating genotypic methods also, but they will never be the gold standard because culture and Kilpauk distribution method and minimum inhibitory concentrations will always be the gold standard. But however, even in Bangladesh, we are now slowly supplementing it with genotypic evidences. There is a concept called resistome that has come up, and this stands true to the name of a mix of genomic and phenomic characteristics. Now, the National Medical Council introduced the LGBTQIA policy recently in education. They said that you have to include an inclusive education. But this has been followed by all our professors for so many years. They've been teaching us about gum hyperplasia and breast cancer and trans men and trans women for so many years. So that is what we need to take. Inclusive education means learning everything that the professor has to offer, in my opinion. Violence against doctors is one topic that we faced when I was in my second year MBBS. And when I went to, when one of the chiefs brought this topic up, they said, ஆமா பேஷண்ட் அப்டேட்டே பண்ணல, ஃபேமிலி அப்டேட்டே பண்ணல, நீங்கள் வந்து அறுவாள்லாம் அங்கே இருக்கும், பார்த்துட்டு போங்க அப்படின்னு தெரிஞ்சு. ஸோ த திங் இஸ், திஸ் இஸ் வாட் they've proven in AIMS Delhi. When I had the fortune of interacting with the director of AIMS Delhi, sir was telling that they were able to reduce to almost zero violence even during peak of COVID pandemic by taking videography of the entire COVID ward and sharing it to the patient's attenders. So the patient attenders were reassured, saying that they could see the patient being alive and being taken care of. The KMC banyan has always been a power of knowledge. It has attracted so many of my old professors here today, and I'm very grateful for that. Everything that is being offered to us is spoon-fed nowadays to us. But it is up to us, all of us students, to swallow it. That still remains in our hands. One more thing I learned from all the faculty, which I'm eternally grateful for, is the ability to learn from everyone. Professors, I've seen them come sit in CMEs and take notes. That was so inspiring for me, and till date I'm very grateful to Arundhati Amma, who's maintained the genetic lab for 10 years. She has taught so much. She's taught so much about genetics and so many things. In fact, I would go to the point of saying she's troubleshooting queen Amma, because she will tell, இதை clean பண்ணிட்டேன், இங்கே வச்சு திருப்பியும் ட்ரை பண்ணு, இதை வந்து நீ அப்படியே ஆட் பண்ணாத, இந்த பிப்பெட்டை நான் அலம்பி தரேன், you try this here and it will work. And because my professors made sure that I'm open to learn from all of that, I am what I am today, and I'm very grateful for that. The most heartwarming experience in this club was that juniors took to this club like fish in water. The moment they stepped in, I would fail in my duty if I didn't mention Sneha, Varun, Mohil, Deepak, Vaibhav and Manish, and the excellent council who are there with me, Yuvakumar, Roshan, Fatima and Sachin Nair, who played a very, very vital role for both today's function and for the club activities the past 7-8 months. I thank my mother and maternal grandparents for making me realize so early on in life that I owe every achievement, every drop of success to all my gurus, to all the wonderful faculties. That means more to me rather than giving back to the society, giving back to patients, giving back to my alma mater or to my juniors. I humbly place this club as a dedication of Guru Samarpanam at the feet of all my faculties. Bharatiyar has always envisioned everything 100 years in the future. He said, Ilaya Bharadathinai va va va, Edirila Valathinai va va va, Oliyanda Natile Nindrerum, Udayanyai Roppave va va va, Kalayiranda Natile Pole, He has told everything that we have planned in the club. Our entire objective summarized in 6 lines in a Papa pattern. I would like to conclude by saying that this is an unedited, from the heart speech, because this club, I believe, very sincerely, has the potential to inspire, motivate, and change thousands of lives. I seek sincere forgiveness if I have made any errors. Thank you so much for the opportunity. Now moving on to the video release. I would like to request Professor and Head of Microbiology, Professor Dr. Ravinder sir, and Professor Dr. Rajapriya ma'am, Head of Anatomy, Omudrar Medical College, to officially release the video that we have prepared for the launch. Ma'am, please, ma'am and sir. And Professor Head of Biochemistry, Dr. V. Meera ma'am, to launch the video.
Thank you, sir and ma'am. A few words about the video. Snippets from the Experts is a series of videos from directors, deans, and senior faculties from prestigious institutions across the country, like AIMS, IISc, IIT, etc., to name a few. One point to note is that these videos have been made specifically for our institution, Govt Kilpauk Medical College and Hospital, by various esteemed members who have made immense contributions in the field of science and medicine. Here's a preview of the video.
Hello friends, my best wishes for the World DNA Day today. I am speaking to you on request of Balaji, whom I met at a recent workshop that we conducted at our center. I was very happy to see his enthusiasm towards research and his interest in learning more than what is routinely taught in MBBS. I was also an MBBS student just like you few years back. However, after my MBBS, I entered into MD in pediatrics, and after that I tried out doing private practice and working in some hospital, but I was not really happy doing the routine work of seeing patients and prescribing medicines, and that is when I came to know about the DM program in medical genetics at Sanjay Gandhi Postgraduate Institute, Lucknow. So I decided, let's give it a try. Although genetics is really a research subject, but there was slowly some progress happening and clinical utility was coming up. So I joined there for a DM program, and during those 3 years at the institute, I learned a lot about how practice should be done based on evidence, how we can use the principles of genetics into clinical practice, as well as it created an interest in research in me. So after that, I decided to join a research institute and follow my career into research. And I can assure you that after almost 15 years of being in the research field, I am as happy as I would have been while seeing patients. It is very important for clinicians, for medical graduates in our country to enter into research field either as a full-time career, or you can take up research in whatever field that you are working. It's not really necessary that you have to become a scientist to do research. All of you will be graduating, then doing some post-graduation, and you will be reaching some or the other hospital or a medical college. Wherever you go, try to do some sort of a research in whatever facilities are available, so that will help you to build your academic aptitude, to help students to come into the research field, and you will surely enjoy it. I hope at least some of you are able to take up research as a career, and we have so many examples of clinicians who have taken a full-time career into research and they are doing very good work. India is really in need of physician scientists, and I hope some of you at least will take up this field. Best wishes for your future, and thank you, Balaji.
Dear students, I understand that you have started a clinical cellular and molecular genetics and translational research lab some time ago. This is a great step to create awareness among clinicians on clinical genetics, human genomics, and also various genetic and molecular diagnostic tools. Looking 60 years back, when I entered the MBBS course, the then available biochemistry textbook described that the function of DNA is unknown and the basis of heredity resides in proteins. According to that textbook, the protein biosynthesis is taking place by replicating the available protein. We have traveled a long distance from that past. Molecular biology is defined as the study of gene structure and function at the molecular level. Molecular genetics is the scientific discipline concerned with the structure and function of genes at the molecular level. It is estimated that more than 6% of all infants born alive suffer from genetic diseases and 1% from chromosomal aberrations. These involve alterations in DNA makeup. To take an example, congenital sensory neural hearing loss is the most common sensory disorder with approximately 1 in every 1,000 newborns. Most of them are due to genetic causes, and there are more than 90 causative genes identified. This reflects the complex clinical and genetic landscapes of hereditary hearing loss. Gene therapy was once considered fantasy. However, thousands of individuals have already undergone human clinical trials. Gene therapy is effective in inherited disorders caused by single genes. Most dazzling accomplishments are severe combined immunodeficiency, Duchenne muscular dystrophy, cystic fibrosis, hemophilia, spinal muscular atrophy, and so on. CRISPR technologies have revolutionized the genetic engineering. With CRISPR, it is now possible to modify, delete, insert, activate, or inactivate any gene as well as fine-tune them. Thus, a troublesome gene can be inactivated. CRISPR technology is often hailed today as the future therapy of genetic diseases. Cell and molecular diagnosis, that is CMD, have become a separate entity in medical laboratory setups and have become an important tool for better diagnosis and treatment. CMD gives insights into the pathogenesis and prognosis of various diseases.
Hello, my dear students. You know that 25th April is being celebrated as the DNA Day. And we start in 1953 when the first DNA report was published. And 50 years later in 2003, the first human genome map was published. And the next 20 years, there are much more technological advances leading to our better understanding of the human genome. And still, we don't know about the exact sequencing of the human genome. They are based on the bits and pieces woven together to build up the human genome map. And in 2022, one of the advances was the gapless human genome sequencing. Still, there are other regions in the genome of an individual which are repeated sequence. Now, these repeated sequences provide diversity to the human population. And that's why when we talk about the human genome, one thing is that there is a one humanity, but multiple genomes. And as a part of the career in the medical genetics, we have to understand that the God has always created something hidden to us. And we have to learn this a long way in the coming years. That is, what is the written things in the genes which can dictate our future. And that's why for interest in the medical genetics is very important, as we know that it is now having the impact on all the biology. I wish you all success for studying the genetics as a unique subject because it gives a much more curiosity, it's elicits your curiosity to the utmost.
Hello students, this is Dr. Akshit Bolur and it's a great privilege for me to address your club, that's the Clinical Cellular Molecular Genetics and Translational Research Club that you have having at the Kilpauk Medical College. I've gone through your activities and it is exceptional that you have done so much of good activities in the last 1 year. On this day, I would like to emphasize upon the need of research in undergraduate curriculum. Remember, it's not about learning what is already known to you, what others have done. It is important that you also do something for the future. So research has to be started, however small, however big it is, does not matter. You will have to take a small topic and possibly research on it and then get into some results which will be of useful to the future. I congratulate the club and I wish the club the best for all their future endeavors. Thank you.
Hey, hi everyone. I'm really happy to be doing this video on for the World DNA Day celebrations. And it's really a pleasure to talk to MBBS students for the future of medical treatment in this country. I think I just wanted to say a few words about what is it that we've been doing in our lab. So my lab works on computational approaches to understand and manipulate biological networks. We also do quite a bit of analysis of large biological data sets, typically which we call multi-omic data sets, wherein we have genomic information, transcriptomic information, network information, and so on. And we solve a spectrum of problems. We tackle a spectrum of problems in this space. Our primary focus is on microbiome systems biology, but I think for the MBBS students, what would be more interesting is our work on cancer biology, where we've been applying a variety of machine learning methodologies to try and understand what are the key genes that drive the progression of cancer in various patients. So we've done this on pan-cancer models, meaning across cancers, what are the key genes, which are called driver genes. So these are genes that specifically confer a growth advantage to the cancer cells. So these could be tumor suppressor genes or oncogenes. I'll not get into all the details. You can read more about it in our work. But there are various kinds of genes. But what is very interesting is that cancer is so heterogeneous, right? So the same gene in one patient or the same mutation in one patient may not drive cancer the same way as it does in another patient. So for instance, a particular gene could be a tumor suppressor in a particular mutation in a gene could be a tumor suppressor in one patient, but the same could be something that causes tumor in another patient and so on. So there are these very diverse etiologies for different cancers or even the same cancer across different patients. And there is so much data that's available now from genome sequencing, from doing RNA-seq to analyze the transcriptome or the various RNA transcripts in every cell. And there are also studies looking at proteomics and so on. And the interactions between various molecules in different cells. So can we use all of this information to learn what could be the most important aspect or the most important mutations that drive cancer in different patients. So this is something that we do, and you can check out our lab website at IIT Madras, or also follow me on Twitter, and our Center for Integrative Biology and Systems Medicine, or our Center for Data Science and AI at IIT Madras, where you can read a little bit more about our work on tackling various interesting problems, including a very recent pipeline that we have designed for a GUI pipeline, where you can just put in sequence data and study a little bit more about what are the kind of mutations that are there in the patient and things like that. Thank you, and I wish you all the best and all success.
Hello, Really happy to be talking to all of you through this video. Thank you to Dr. Sudharshanan Balaji for taking this video and having me talk about some of the research that my laboratory does here at the Indian Institute of Science, Department of Bioengineering. What we are primarily interested in is to look at how to modulate immune responses using drug delivery systems, biomaterials, maybe develop better vaccines or develop better ways to modulate immune responses for a variety of diseases. And we have a clinical focus in our research. So we do collaborate with many clinicians, do clinical studies. In fact, one of our most forward-looking ideas is to figure out whether we can help people with diabetic foot ulcers. A large proportion of diabetic individuals, so somewhere between 10 to 30%, end up getting these foot ulcers. And many of them, with the current state-of-the-art treatment technology, still do not heal their ulcers. And so through research in our own laboratory and through clinical studies in collaboration with clinicians here in the Karnataka Institute of Endocrinology Research, we have determined that there are certain immune markers that seem to be upregulated in individuals that do not heal their ulcers. And so we've been designing new bandages that can be potentially immunomodulatory in some way and help heal those ulcers faster. So part of it is clinical research where we identify these biomarkers. Part of it is laboratory benchwork research, making these bandages. And we are also doing preclinical research where we test some of these bandages in animal models of research before we can take them forward for clinical tests and trials. Similarly, through some of those studies and through other interactions, we have started networking with clinicians on chronic kidney disease. We've also been working with individuals who work on gliomas, clinicians who treat gliomas, and trying to understand how immune cells are altered both in the brain tumor microenvironment as well as in chronic kidney disease in circulation, and those individuals who undergo dialysis. And the hope is that all of this research will lead to a better understanding of how immune cells change in these individuals and whether specific treatment strategies, maybe existing drugs but not used for those sort of indications, or newer therapeutic modalities can be identified to promote health and well-being of individuals with chronic morbidity. If you have any questions, I'm happy to answer them. You can write to me. My email address is siddharth@iisc.ac.in. You can look it up on any of my department websites. And wishing you all the very best and looking forward to seeing many of you becoming more interested in research while doing your clinical practice. I think clinical research is the way forward. Thank you and have a good day. Bye.
Treasurer Jayaganesh Esra from the 2020 batch. We now request the faculty in charges to kindly administer the oath to the new council members.
I solemnly swear to do my very best
I solemnly swear to do my very best
in performing the duties
of the student council
of the clinical, cellular and molecular genetics and translational research club, Govt Kilpauk Medical College, as the
Vice President,
as the treasurer,
As the club joint secretary,
Secretary.
I will proudly lead my peers and all members of the club and openly listen to their concerns throughout the academic year. I will put forward my best efforts in carrying out my roles and responsibilities. At all times, I will be an exemplary role model for all to see. I will strictly follow the general rules of students' conduct expected of me in this esteemed institution. I will make use of this opportunity given to put in efforts to make this club reach greater heights and to bring pride to KMC.
we now request the faculty in charges to pin the badges and install their posts.
all the best.
Thank you. Acknowledging all the good we have now is the foundation for all abundance in the days to come. This occasion would be incomplete without acknowledging the efforts of all the people who have worked in synergism to make this day a success. They deserve our unfettered gratitude, and hence I would like to humbly invite the joint faculty in charge of CCMGTR Club and Head of Department of Microbiology, Govt Kilpauk Medical College and Hospital, Professor Dr. Thyagarajan Ravinder sir, to kindly come to the podium to deliver a formal vote of thanks to this benevolent academy. Please, sir.
Respected professors, dear friends, very good morning. And as the Mime team, they said no life without genetics. It's very true. There's no life without genetics. And the basic of genetics which Mira madam quoted in the morning is DNA. Except in RNA viruses, all other living forms, they require DNA for replication to transmit its characters, both in hereditary as in as variations. So it is basic unit of life is DNA, and with that, the club has come to this extent. What Sudharshanan Balaji has told about the various events through which the club was initiated and how they cherished with the help of faculties to come to this level with so much of dignitaries honoring and praising the club in their videos, and few of them even wants to put their students or learners into the club. That is a big achievement. In this occasion, I take pride and honor to thank our dynamic Dean sir who has come here in spite of his busy schedule and also gave his advices for the club. And two important things from Dean sir's speech today about the gene therapy and getting extramural funds like DPT fund, which one of our candidates has received, and many of the ICMR fund. But we have to go for more ventures, and at this point I again repeat, your alumni is ready to help you. They conveyed to me for research, they are ready to fund and also help you go abroad and present in various forums. I am repeatedly telling the members as well as few of you have requested also, please present whatever you have made. National level presentations, all of you have made and achieved some prizes also. And this year we need to take it to the international level with the madam's guidance. Our members can go and present in the international forum, as well as he has not read about any publications. I'm little worried about it. Only publications will take club's name, the individual's name, as well as the institution name to world level. Whatever reputed institutions you put forth in the videos, like my own alma mater, Stanley, as well as JIPMAR, both they are in a different level as CMC Vellore or MMC was, but now it has little setback. It will again come back. AIMS, everyone is only based on their publications. If you take the patient load, our institutions are leading than any others in the country, but publication wise we are little behind. So this elite group of audience will definitely fill that gap and put KMC as well as all our related colleges in the map of the world. This is my ambition, and hope you will all fulfill. And our VP ma'am and medical superintendent ma'am are always encouraging for us, and anything they guide us in the right direction. I thank both of them who have come to this function and honored us, and also our RMO madam. In spite of her busy day today, she has come over and graced this club, and various professors from other institutions, actually from our institution only, Rajapriya ma'am, as you all would have been trained by madam. I thank madam for coming all the way, Anjana ma'am, and Gita ma'am has already entered KMC, Ratna Kumari ma'am, and Balamina ma'am, very much connected with genetics because she is dealing with rheumatological cases. Only genetic background will help curing many diseases, and one of them is rheumatology, madam. Thank you for coming today and gracing this occasion. I thank Sugati ma'am and Teres Mary ma'am from my own department. And always I used to tell whenever I come to the club, it's not alone human genetics, it is bacterial genetics and protozoal, then fungal, and viral genetics is unique. They have only one component, either DNA or RNA. So few of you can venture into those areas and try doing some research. And Mime was excellent, right from Watson and Crick to COVID, coming all along with microbiology, Griffith experiments, and my favorite experiments of Mendel. Even HLA meeting I was mentioning with the PowerPoint. even now, every day, right from my school days, I cherish those experiments by Mendel because it paves way for explaining all the disease. It gives explanation for all the disease with little modification even for the bacterial genetics. With all this, I thank all other faculties who have come here. Our nephrology Balasubramaniam sir and Tirunavakar sir has come. OG HOD Vanitha madam has come. So I thank everyone and special thanks to the founder members Sudharshanan, Balaji and team, and the new members who have been inducted now. The new team, definitely they will take you to greater heights, and each and every one of you to have to do research. And one more point I need to mention here, in the forthcoming national education policy as well as state education policy, more than 60-70% importance is given for research. They have given importance for research at school level, so you can see the impact at the higher education level. Ultimate, it is going to be in the world of research, so please take it up. And I thank every volunteer, all the participants. Our, as he told, Arundhati Ammal is the backbone for Gene Lab. I fully accept and appreciate her work because many endeavors behind, and many times I tell it's not alone the book education, they have learned by themselves. So I thank each and every one who has contributed to this club. Thank you one and all. Please achieve great laurels and do things, go present internationally and also publish in international journals. Thank you one and all.
I must thank the special thanks to the judges from our department, from pharmacology, microbiology, anatomy. Really, it was a good integration. I must really thank, because it was after the classes, after 4 o'clock. So everyone stood and they really enjoyed it and they judged it. And certain competitions they were not able to judge, like they have to give prize to all. Sugati was telling that everyone presented a very good rare case presentation. So it's a nice start. All the best.
Thank you, sir and ma'am. We would also like to inform that posters of various research activities undertaken by the club have been put up on the left. We request you to kindly look at the same. As we near the end of this session, we request Professor Dr. Meera ma'am to give us an opportunity to fall in love with her voice all over again by rendering the national anthem. I request all of you to rise for the same.